No, I don't want to switch. I did it again.
Oh, even that's doing it. Okay, there's that. Because we were already logged in. I wonder if it's the actual portal part that's sent to you. Sorry. I have no idea.
Recording in progress.
Sorry. All right, we'll try that. Cool. Just pretend like that's not there because I can't make it any smaller. But this will be better than. All right, sign-in sheet.
Mostly all here.
So because all we're doing for lab today is the review, which there kind of is one posted. I couldn't get the one that I did with my Monday class to post. It's like still on my computer up there and I have to post it. Believe me, my day is not done because I still have to set up the exam. But anyways, there is one posted, a review, if you just don't want to do it with us. However, if you want to do it with us, I'm going to do it just right after lecture, right in here. I'm not going to go upstairs because why? It's just cold, still cold, and just nasty and cold. So let's not do it. All right. So any questions about anything, the exam, what you're studying? All right.
Off topic, but just before you get into that, I'm just curious. Did you or has anyone figured out what's going on with the cadavers in the anatomy lab?
I'm still kind of working on it. We have to order like special tests for it. When I do, I'll let you know if I know anything. Yeah. Okay, so chapter 11, I don't know why this can't just be full screen. Chapter 11 is talking about, no, it's not easy. It can't be easy. Why would it be? It's talking about microbial control. And microbial control like refers to essentially, making sure things are clean. When you guys disinfect the lab tabletops, that's what we're talking about, right? When you wash your hands, that's what we're talking about, okay? So here are our 4 main terms associated with microbial control. We have sterilization, disinfection, antisepsis, and decontamination. Now, I don't know how many of you feel like you have just a completely great handle on note-taking and being sure that you are writing down or, typing or whatever it is, the things that you need. But if you don't feel confident about it, I'm going to walk you through it anyway. So here we go. This is from this slide where I would start. I would write down my terms and what they do. If I were to write down in my notes, sterilization kills all. What we saw in the previous slide, it's way slower on that screen on mine. Okay. What we see in the previous slide, sterilization, the destruction of all microbial life. Kills all. Are we in microbiology? So what were we talking about if it kills all? Microbes, right? So you don't need to be writing on that much for that. That's just beginning. So look, for disinfection, it destroys most. That's not all, right? And most of the time, it's the endospores that are going to survive, and we'll talk about that too. Then we have antisepsis. I should carry this around so people can hear me in the recording later. Okay, so next we have antisepsis. It's also called degermination. In case you can't tell, I'm not going to refer to it as that ever, but it is. Antisepsis is talking about disinfection on living tissues. And then decontamination, it's mechanical removal. And so now I would tell you that, what do I mean by mechanical? I mean by scrubbing it off. When we're trying to get the microbes off, which is often like detergents and things like that will help us with that. Or if we are like separating from like a filtering like a liquid out and we've separated them, but literally physically removing them from the situation. Whereas other things like the, disinfectant that we use in lab, it kills it, or hopefully it kills, but it kills most of the things, but we're not actually removing them, right? It's just the whole point is to kill them. So that's what we're talking about there. So now when we come back to this, again, sterilization kills everything. Disinfection kills most, but that's not everything. Antisepsis on living surfaces, decontamination, mechanical. All right, so here is a great chart that we can refer to later on when we have gone through everything and see what sterilizes and what just decontaminates or disinfects or something, right? So the reason this is important is because most of the questions I'll ask from chapter 11 will be, which of the following things in this list sanitize, or does blah, blah, blah sanitize, true or false or whatever. So sanitization, well, sterilization, I guess, is the more important one. Does it sterilize? And then if it doesn't, you can usually figure out if it's disinfection, antisepsis, or decontamination, just based on how you're dealing with it, the sample. Okay. I have a little note. Let's see. I forget what it says. You're going to see my face pop. It's me as a kid, by the way. little baby Lauren, 2-year-old Lauren. All right, sterilization will only be achieved, just to remind you guys, it's only going to be achieved if we follow the instructions, right? So if you're not treating things long enough, it's not going to sterilize. It's just going to only kill some of the microbes. So just because I tell you that it sterilizes on here, it doesn't mean that it always does. It just needs to follow those rules in order to get that far. But the other ones can't get that far, even if they try. I'm going to get rid of this. Collapse. Okay, that's not what I wanted to do. All right, so to look at resistance of our microbes, the ones at the top are, why is it doing that? The ones at the top are most resistant, and the ones at the bottom are least resistant, or as we learned from Microbial, most sensitive or susceptible. So up at the top, we can see I have highlighted up there, it's bacterial endopores. And there's a thing above it. You guys see that, the prions? I didn't put them on the list. I think it says it on the box, but I'm not going to open it because they're just really rare, really uncommon, and they require exceedingly high temperatures and situations that we don't usually use to clean things. It's just extreme in order to contain them. So I don't list them typically. So I would typically think that the most resistant, all the stuff that we are designing to be used to clean things is usually targeting the endospores. And that's what we would consider to be sterilization. If you get rid of the endospores, everything below it will die also. And you guys remember, those are the hardy, the dormant ones that are meant to survive harsh conditions. All right. Like I said, bacterial endospores are the most resistant, and the goal for sterilization is to get rid of them. We just don't see prions enough to care about it. So if we kill endospores, it'll kill everything. Going into a little more detail on these, sterilization, again, kills everything, removes all viable microorganisms, viruses, endospores, all of it. Anything that's been subjected to that treatment is called sterile. And of course, we want to sterilize our surgical instruments, syringes, and packaged foods. And you guys might even be thinking, like, whenever you get like a syringe out, if you're working at any sort of like a vet clinic, or if you're at a hospital, whatever it is, they're usually packaged. Yeah, the new ones will be. And the needles will be as well. Typically, those probably were not sterilized in the way that you think that they were. Most people think that, oh, well, everything gets autoclaved, but nope. I'm gonna tell you what actually happens, because it's kind of interesting. So if going forward, I'm going to say that this kills some blah, 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 or this kills most blah, 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 then that means it doesn't sterilize. So you can just say it right off the bat if you see that, because it has to be all. All right, so like we said, disinfection, you're not killing the endospores. That's probably the most important aspect of it. And we usually use that on inanimate objects because if you're putting it on living tissues, it is antisepsis. Sepsis refers to when you have microbes growing on or in tissues where it doesn't belong. And asepsis is preventing that infectious material to get into the tissues. So when we talk about aseptic technique, it's any technique that we're using to prevent contamination where it shouldn't be going. So antisepsis is a type of asepsis. It falls under that category. So this is the application of chemical agents called antiseptics. Okay, so that's not a weird word. Now we're getting into the realm of stuff we recognize maybe. An antiseptic is used on living surfaces to kill vegetative, not endospores. It's never going to sterilize. So here's how we updated it. We already wrote our terms down. That was in the black writing. Now the red sterilization, we're going to even include the endospores. We'll kill those as well. They are the most resistant ones. We've made a little note about that. If it's disinfection, we added on not endospores, not killing those. And then antisepsis refers to even including the aseptic technique. And antiseptics fall into that. Decontamination, we haven't really talked much about it yet, but it's just going to be, again, mechanical removal. We remember these terms, cytal and static, right? Cytal is going to kill, static inhibits, but doesn't kill. So we already know these ones. Based on like the beginning of it, if it's germicidal or microbicidal, it could affect any of them, right? That doesn't mean that it sterilizes necessarily. It just means that it targets all groups somehow at some point. If it's sporicidal, though, it kills endospores. And so what would that mean? That in particular would be sterilizing. It has to be. All right, cool. Static, we already know. I'm not going to really worry about this one. You guys know static. Why is it so slow on that screen? We're not killing with that one. Okay, here's our decontamination, our mechanical removal. This is going to be physically removing, usually by scrubbing, washing, filtration, but it also includes coughing and sneezing. That's your body trying to physically remove, contaminants from you. So that makes sense when you think about it that way. Hey, that's your body doing that, decontaminating itself. So now I can add that stuff into there. Decontamination, an example that we might know is scrubbing, like we would have in surgery. And then cytal, however you want to look at it, cytal will kill, and then static doesn't kill, just inhibits. Ready. If you are looking at things as far as picking your means of removing microbes, right? You want to remove microbes, but do you need to destroy the endospores, or is just the vegetative okay? Do we need to reuse it, or are we going to use it this one time and then discard it like we would with a syringe or something typically? Does it need to withstand heat or pressure or radiation or chemicals? Is it able to withstand those things? And is that suitable for that application? Will it penetrate as much as you need it to, and cost effective? So think about it this way. Some of these guys, and I'll show you, but some of these guys are like gases, right? Or we'll talk about the autoclave that uses high heat and pressure and steam. So would you treat someone's arm to prep them for an IV with autoclave or gas? No, this is absurd. So that's what this is talking about. There is a lot more out there than just the things that you are thinking about maybe at that moment, right? So we're going to divide our things that we may need to keep microbe free into 3 categories. Critical devices, which will come into contact with sterile tissues like we have with surgery. Semi-critical devices that are going to contact our mucosal membranes. This is usually going to be like our respiratory equipment and thermometers, things like that. Non-critical medical devices, they're either not going to touch the patient or they're only going to touch intact skin. So door handles, typically the bed, unless there's some sort of oozing or something. or any of the like machinery around, keeping track of heartbeat and things like that. So now we have to think about things that could affect killing our microbes. How many microbes are in the area, nature of the microbes that are there. Are they endospores? That would be a concern. Are they forming any biofilms? Because then we have to treat those completely differently if they've got like basically layers and layers of bacteria. Whatever the temperature and pH is around us, the concentration of the agent, temperature and pH might actually affect your agent. That's why that's a concern, okay? The concentration of whatever you're using, the mode of action of whatever you're using, and whether it can be interfered with by other things in the area, like organic matter, which would include pus, blood, feces, mucus, and urine. So yeah, it's not as simple as just autoclave everything. Like we said, with looking at arms, that's not going to work out very well. So you still have to prepare those and remove the microbes. Okay, so this is how they work. They're going to either attack cell wall, membrane, DNA and RNA synthesis, or proteins. And that's pretty much how the physical ones and the chemical ones are going to work. And it's also how our antibiotics are going to work when we get to those. They have one more category, but other than that. So now we've updated this and added on what we're getting ready to see, our cell wall and BS like that, right? All right, just to show you guys, this is all the notes that I would have taken up to this point. Okay, just be clear, I literally went through this slide by slide and made notes for that. Okay, so cell walls, we can block them by blocking the synthesis, making them, or by destroying them. So that's two ways we can look at it. Once we destroy the cell wall, it's easier to lyse or break open the cells. And then some things that work pretty well with disrupting cell wall tend to be the things that are also good at disrupting the membrane. And that includes detergents and alcohols. Kind of funny that like we use hand sanitizer so much, and today is the day that you guys get to learn about how it works. And that's kind of cool in a way. I don't like this chapter personally, but it's not as cool as the other ones, but it's a little bit informative. All right, so cell membrane also includes the viral envelope, right? Not all viruses have it. If they don't, they're called naked. If we affect the cell membrane, we will have loss of selective permeability, which is essentially saying the water could move in and out, but other things had to have channels and things to be controlled. So if we break this apart, we can't have selective permeability. We'll lose vital molecules, and damaging chemicals can get into those bacteria. All right, so something that affects our cell membranes are surfactants. Detergents are surfactants. And these are molecules that have a hydrophilic and a hydrophobic region. Okay, dude, I don't want that. Thank you. A hydrophilic and a hydrophobic region. You guys remember about these, right? Hydrophilic, water loving, hydrophobic, water fearing. And if you have both going on at the same time, it gets its own term, which is amphipathic, right? We've heard it. And there's one molecule that we know pretty well at this point that is amphipathic, and that is the phospholipid in your membranes. So what I'm telling you is that these surfactants mimic a phospholipid. They get stuck in between the phospholipids and cause it to basically the membrane to fall apart. There's a picture of it here. Come on. The little teal guys in this picture, these are our surfactants, and you can see them. We call it intercalating, but it just means squeezing between the other molecules here. And then breaks apart the integrity of that. So yeah, it's not good for the cell. All right, next we have the ones that can affect nucleic acid synthesis and proteins. We're looking at inhibiting proteins. We can inhibit the actual manufacturing of proteins by stopping the ribosomes. That's a pretty common one for our antibiotics later when we talk about those in the next chapter. But we could stop the ribosomes to inhibit the translation. There's another way we can deal with the proteins. We'll get to that in the next slide. But then we have agents that impede transcription or replication. I don't know why that says transcription of DNA replication. That's not what that means. But DNA replication, making copies so you can make new cells, or transcription, taking your DNA to RNA, which you have to do in order to make proteins. So you target any of those steps, you can stop the cells at least from growing and creating new colonies and things like that, and potentially killing them. All right, so this is the other way we can take care of our proteins. It's denaturation, and it's a term you guys know already. You know that we talked about this with DNA and especially with PCR, which in case you guys don't remember, that's the first step, right? DNA is denatured because of the heat. Same thing can happen with proteins. They're still depending on a lot of hydrogen bonds. So it can be affected by temperature or even pH, because remember, that's going to affect the H pluses in the area as well. So we can denature that way, thereby breaking those. Usually it's your hydrogen bonds that maintain the secondary and tertiary structure. Usually we'll do this with moist heat or certain solvents, like would include probably glutaraldehyde, for example, and then phenolics that are based on phenol. Don't worry, we're coming around to all of these. And then metallic ions will bind to active sites. So these are just examples for right now. Believe me, we're coming back to it. Okay, so moist heat will denature, just like whenever you cook an egg. No going back, right? We talked about that. So here we go, different levels of denaturation. You can have complete denaturation, or you can just change its shape and it can't perform its function the same way. Both of these are going to be denaturation because it can't function the way it's supposed to. So we had the top part all up there, and the yellow was already taken care of. So we have this down here in our black, tape or whatever you want to call that, the cell wall and etc. I made little notes there for what you need to do in order to affect them. Cool. So that's done. That's one sheet done. And now we're going to move on to physical control, and then we're going to go into chemical. Okay. So our main modes of physical control, definitely heat. Physical control, heat is like the number one. Then we have radiation, filtration, ultrasonic waves, which we're not going to talk about at all. By the way, we'll talk about osmotic pressure, which is talking about like packing in salt. Okay. Remember osmosis, and that has to do with water traveling across. And you pack something in salt, it creates osmotic pressure. Then we have cold. All right, so I've made my little notes on those, getting ready. I had to space them out quite a lot more than that, by the way. So I had a lot more to write than I thought I would. Okay, so we'll start off with heat. This includes any hot water at all, boiling water, and steam. Okay, that's the most ones to think about, but anywhere from 60 degrees Celsius up to 135 degrees Celsius. This is the temperature that we would need to be at in an autoclave to get rid of those prions. Okay, so it would be much higher than what we usually do is 121 for most things. So anyways, you can use pressure to control your temperature a little bit more easily, get there faster, to make it more effective with the water. The water is actually important for causing that coagulation and denaturation effect. So it's why moist heat is more effective than dry heat. Because dry heat, when you put something, you burn it or you put it in an oven, whatever, anywhere from 160 now up to several thousand degrees, right? This is going to dehydrate the cell, and when the water isn't there anymore, it can possibly increase the stability of proteins. It's almost like it's preserving them. So you just have to get really hot and like actually burn them to ash, oxidizing them and burning them to ash. So here's a comparison between moist heat and dry heat. If we look at 121 degrees Celsius, which is what we use for autoclaves, and then putting something in for 15, maybe 20 minutes, commonly done for autoclaves. Then we have the dry heat. In order to sterilize something at that temperature, that same temperature, you're going to have to go for 600 minutes. That's 10 hours. That sounds like a nightmare. So how do we make this faster or better? You're going to have to probably increase the temperature if you want it to work, and not everything's going to be able to withstand that temperature. At the same time, you might have things that can't handle that moist heat or the pressure from the autoclave. So if it can't handle moist heat, that would be certain kinds of metals, for example. So you might have to just sit it out with that dry heat. So talking about thermal death, killing things with the high temperatures. You will destroy endospores as long as you're at temperatures above boiling. So if that doesn't tell you about boiling, it should tell you boiling doesn't sterilize, right? So don't, by the way. Vegetative cells will vary in their sensitivity. It just depends on what cell we're talking about. So that's the living multiplying ones. Yeah, we want to get rid, the goal is to sterilize this to get rid of those endospores and everything below it. So if we're looking at trying to kill things with heat, there are two approaches you can take. You're going to figure out the thermal death time and or the thermal death point. So time and point. Thermal death time, essentially the time it takes to kill. So you're going to have a set temperature. How long is it going to take to kill all the microbes? For the second one, this per thermal death point, you get 10 minutes to kill all these microbes. Tell me what temperature you have to do it at. That's what that is asking. What temperature? Okay, boiling water is for disinfection, not sterilizing. It's pretty effective for non-endospore forming pathogens. So if you're treating things at home, like bottles for babies, and then utensils and things like that for from people who are sick, whatever, it's good for disinfection for that. It's just not going to get rid of the endospores. Next, we have pasteurization. You guys probably heard of this. This is what they do to your milk, or should be doing to your milk. It is disinfecting. It is not sterilizing. We already know that, because if you leave your milk in the fridge for a month, it's going to get chunky. And even if you don't pop the top, right, it'll get that way eventually. And that is because there's still microbes in there, and they are fermenting in that milk while it's sitting there, even in the cold. So that's pretty crazy. But yeah, obviously leave it out, it's going to do that faster. But pasteurization just cycles temperatures. So we go from kind of high, look at that, 71.6 Celsius. Boiling is 100. So we're like well below that. So going to that, just for 15 seconds, we'll hold it and then let it cool back down to room temperature. And once it's back down to that stable temperature, back up again. And they'll just do that over and over again until they've gotten rid of most of the pathogens. And so this is really pretty ineffective at removing everything, but it was okay. It's okay enough. It maintains the integrity of the liquid. We're not destroying proteins that are in the liquid, for example, like we have in milk. Right. So it's not sterilizing. Right. Now, if we want to sterilize something with moist heat, because I keep telling you that that's like one of the preferred methods, we're going to use an autoclave. In an autoclave, we're going to have steam and pressure greater than an atmosphere. And our atmosphere right now is 1 atmosphere. That's our pressure here. So we can use temperature around 15 psi, pounds per square inch. And that will let us get to 121 degrees Celsius. And we can only have to hold it for 15 to 20 minutes to sterilize what's ever in there, so that we can get everything nice and sterile and reusable. We'll do this with instruments like surgical instruments if they reuse them. This is pretty common for most of what they're going to use in a vet clinic. If they're going to reuse it, they'll be autoclaving it, usually in a tabletop autoclave. So this is a standing autoclave. I guess they would use these too for dental instruments, right? I mean, I guess, yeah, I assume they would. Yeah, I don't think about it because they always talk about this other stuff. I don't, yeah. So we have standing autoclaves, which are like they can be pretty big. The one that we have in like our lab, we don't have it in our lab ourselves, but in that prep lab, the person that makes all of our media autoclaves all of our stuff. And it was liquid at that one point, but now we let it cool into solid, and that's how we get it that way. So it's sterile. But anyways, it's about the size of like maybe the chair, like as far as area that it takes up, like the actual opening in it. The ones that I used to use at my last job were about this opening that you would push these into, about the size of a little wider than this table. And they have 4 of them. You would roll things into it, and like it was just crazy. It was crazy. And like you'd pull it out, you have to wear the gloves because it's too hot, because you put the whole cart in there, and then pull it out and it would be hot, and you need like the special gloves, and like even then it would sometimes be too hot. And you have to let it sit there. You can't touch it until it's like cooled down. But yes, those can come in tabletop versions as well. They're not always going to be giant like that. Oh, I forget, I can't click on that one. Okay. All righty, incineration, obvi that this would be sterilizing. Yeah, we're gonna burn things to ash. Let's do it. So this is dry heat now. Dry heat, we're gonna incinerate, literally burning things to ash. We do this with our Bunsen burners, yeah? Our Bunsen burners are gonna reach 1,870 degrees Celsius at its hottest point. Other furnaces and incinerators will get anywhere from 800 up to 6,500 degrees Celsius. And if you're burning bodies at a crematorium, you're probably going to be at that higher end. It's crazy to think about that. Then we also have the hot air oven. And yeah, it's just like your oven at home, especially if you have a convection oven, because it helps that air circulate and be the same temperature throughout. But yeah, it's just got hot, it's got coils. And then you put it in there and it's dry and you let it go for 2 to 4 hours. And that 150 to 180, I believe it's roughly in the range of how we would normally be using our ovens anyways. So it's pretty, you could do it at home, but not everything can handle the sitting in that dry heat for 2 to 4 hours. But yes, you can definitely sterilize with both of these methods. So I've updated these. I am highlighting in this with that weird yellow color, anything that sterilizes. So I updated all that and made any notes if I needed to remind me what thing will do what. So I put even pasteurization, alternate hot and cold, and I put some terms down there: thermal, death time, and death point. I know it says ultrasonic waves here. That's going to go away in a little bit. Cold. It's usually used to preserve your foods. It's also pretty beneficial for preserving the microbes. So they're usually not going to die. If they are kind of delicate, they might die from the cold temperatures. So again, this says some, you know that it's not going to sterilize. If you are putting your chicken into the freezer, it's not necessarily going to kill your salmonella. You're just going to delay that experience for later. So, so just slow down the activity even to a stop. And we use this, like in science, we use at -70 degrees Celsius down to -135, which is one of your liquid nitrogen tanks. Like we would have human cells contained in there. It's what they did at the transplant lab. Anyways, it can preserve your bacteria that way. So you can guarantee that they're going to come back to life. So yeah, obviously that is not going to sterilize. It's used to store them. So psychrophiles, these are guys that will thrive at cold temperatures and even make toxins in those cold temperatures. So they prefer it cold, so they can be an issue. All right, next, our dry heat. We have desiccation. This is dehydrating. Sort of speaks for itself. Like, I don't know. You can preserve some pathogens, but usually it's like we dry out your beef jerky, for example. You hang it out there and let it dry out. If it's preserving the jerky, it's probably preserving the bacteria. But if you eat it, it's not giving it a chance really to like grow and become a problem. So it can be good enough. Lyophilization. This is using a combination of freezing and drying. We usually will take a sample, put it in a centrifuge that will spin it fast and have a vacuum to suck the air out. That encourages the drying process. And then we will also have a freezing temperature all at the same time. And that will create that whatever it is in there into a fine dust. If you've ever seen any of those like glass vials that you have to like go in through like the rubber at the top and add in like a liquid in order to resuspend it, I kind of like them. They're sort of satisfying how like quickly that they'll reabsorb the liquid, but that they were lyophilized. And all of the samples that you guys use in lab were lyophilized and reconstituted. So it's cool. So freezing and drying at the same time, it's good for preservation, but not for really killing. All right, let's get into some different things. Radiation. Let's get cool. Let's get cool now. Okay, radiation. This is where I will mention to people, and I've mentioned before, and I'll mention it to you guys because I'm going to bring up Chernobyl here in a minute. I am old enough to remember it. Okay, so I was very little, but I do remember it. Okay, but yes. So radiation. This is the same radiation as what we're talking about coming from like Chernobyl incidents. Okay, it's all the same. It's electromagnetic, and depending on how active that radiation is with the molecules, the waves, I don't say attacking molecules, but energizing them, determines how much damage they'll do. So we've got two different forms of radiation we're going to talk about, but it's going to include things like gamma, X-rays, and ultraviolet, which you guys know. Anytime you're exposing something to radiation in order to treat it, remove microbes, we would call it irradiated afterwards. This irradiation. And ionizing radiation will cause very severe catastrophic mutations in the DNA, whereas non-ionizing causes DNA to form abnormal bonds, not catastrophically so. So let's look at this top one up here. This thing, this gray here, that's a barrier. And this whole mess on the side, whatever that is, I don't know what it's supposed to represent, like coils or something emitting the radiation. I don't know. But you'll see in the top, that's ionizing radiation. It'll go through barriers. Now, X-rays are tame. They're pretty tame. It like goes UV and then X-rays and then gamma. And X-rays, we already know that if you're around them regularly, you should probably be wearing a lead apron. The lead will stop the X-rays. There ain't no amount of lead in the world going to stop a gamma ray. So just saying. That's why you get that meltdown at Chernobyl, and they have that elephant's foot, which is like where the source of most of the radiation is now. And you can get exposed to radiation well far away from where the elephant's foot is. And it is still an issue in that area, the contamination with the radiation. I don't know. So ionizing, bad, worse, the worst, okay? Non-ionizing, this is your UV, okay? And that one, we put a barrier up, you like through any wall, you're not gonna get UV through it, right? It might get through glass, and some of the treated glass, it might not. So it's pretty weak as far as that goes, but that's also why it doesn't create as severe of damage to the bonds that it's treating. All right, so let's go on a little bit more about how they work. Ionizing radiation will sterilize things without heating them. We call this cold sterilization. You remember those syringes and needles that I talked about? That's how they're sterilized, ionizing radiation. They put them in little packages. You can put them in there. It's not going to melt the plastic or anything, not even in the packaging. And it penetrates anything. So it can go through all the plastic and the metal of the needle even, and completely sterilize that whole thing. It's really useful for that, for disposables. And it's pretty useful for certain foods as well that they'll want to sterilize. So yeah, so our ionizing radiation, gamma ray machines, X-rays are ionizing. They're just on the lesser end. And usually if you're going to sterilize, you're going to use even more powerful ones than the ones we have in diagnosing things. And we have cathode ray machines, which you don't really hear much about that, but so yeah, cold. Probably, yeah, because it can literally just, radiation, you don't see it, yeah, and so it just goes through the whole thing and kills any potential microbes that could be there. Like you don't want to be in that when it does that, but it works well for those guys. So what ionizing radiation will do with our X-rays and our gamma rays, it will cause electrons in the outer orbitals of your molecules anywhere in your body, your proteins, your DNA, doesn't matter, and cause those electrons in the outer orbitals to be ejected. So you guys remember how whenever sodium donated its electron to chlorine, right? So when it ejected that electron out and it became positively charged, it became an ion. So we're creating ions all throughout the molecules in your body where there shouldn't be ions. Like that molecule shouldn't be an ion. And that's why we lose those bonds that were made, because those covalent bonds holding us together, they have to have sharing of electrons. And somewhere from ionizing radiation, we lost them. We can't share them anymore. So that's how that works, and why it causes so much damage and can lead to catastrophic problems with the body from like just one exposure to high levels of radiation. Like those guys, if you guys haven't heard about it, the guys that found like that, I don't even know what it was. I don't know what it was, some sort of reactor fuel or something out in like, it was like something from the Cold War era. And they found it while they were hunting, and it was warm and it was cold because Siberia. And so they like took it with them and like kept it near them while they were like going hunting so they could stay warm. And the guys that were handling it the most ended up dying within like a day. just from like, it just, your body can't anymore. It doesn't work at all. And you'll see one of the more common symptoms that you'll have is just your skin falling off because it just doesn't stay connected because the things that hold it together, they're not the things that hold it together anymore. They just don't work anymore. So it's pretty crazy to think about that, but yeah, it's nuts. I think radiation is pretty fascinating. I don't want to study it. That's not a thing for me. Anyways, non-ionizing radiation. This is a more common one. This is UV. We know that that's radiation. So non-ionizing, these will excite atoms and get them to a higher energy state and then cause abnormal bonds, which I'm going to show you in a moment. But first, we're going to talk about that. UV radiation, I don't care about the nanometers, but hey, there's certain ranges that counts as UV. And we will use that sometimes to sterilize surfaces. You can sterilize surfaces, but it can't penetrate, not like ionizing can. So you'll only do that for surfaces. Like you'll have sometimes something that, like you see people working on cells or bacteria or whatever in those special hoods, and then they'll turn on a UV light. That's to sterilize that whole area without having to use too many chemicals and stuff. But anyways, it'll work. It's just, you have to let it sit there for a long time and it's not going to penetrate anything to any, effective rate. All right. So UV radiation, this is what it does. They are called thymine dimers. That's what you guys would need to know for that. Thymine dimers. What that means is, nice picture here, but what it means is if you had pretend like that's an A, T, T, C, G, A, whatever. And then this was T, A, G, C, T. Cool. And then these guys are supposed to be bonded across from one another, right? Easy enough to think about. Now, with our thymine dimers, pretend, what happens is we don't have that across binding happening anymore. Instead, these guys bind together next to each other. And you can imagine if you are going to make copies of that DNA, or if you're going to make any sort of transcript from that DNA, that would cause some problems because those T's aren't going to want to help you bring in an A. They're doing their own thing. So it'll mess that up and that'll cause some serious issues. That's why when you have UV damage, you can progress into developing melanoma, cancer, because the cells at the surface of your skin having this happen to them. Even if you wear sunscreen, this is probably going to be going on in your cells. If you guys are going out without sunscreen at all, which I do, whatever, I'm not judging, but if you don't, then you're having this happen for sure. And don't be too worried about it though, because your body has a repair mechanism in place. It's evolved to learn how to deal with it. And so if it catches it in time, it will cut out those messed up areas and replace them with what they should be. So no worries there. But yeah, if you get too much sun, it can't keep up with it. If you're getting sunburns and stuff, you need to watch it. Right, so it creates, essentially the UV is going to create the free radicals causing those damage. I don't know what else to say about it. This isn't that interesting now. Well, so I've updated my little thing with the radiation guys and made my little notes about not being able to penetrate or penetrating, and then expelling ions versus thymine dimers. Yeah, they both can sterilize. I did make that note. Okay, good. All right, decontamination. Now we're finally getting to this one. Decontamination, mechanical removal of the microbes, so physically removing them. So here we're talking specifically about filtration. It can sterilize if the pores are small enough. If the little holes that things have to go through in your filter are small enough that not even the smallest viruses can go through it, then you can sterilize it. Are you killing those microbes? You're not killing them, but you can have sterilized liquids. Anyways, that makes sense, right? This can be used to treat liquids or air. So air, that would be like your HEPA filters. All right, next we have osmotic pressure. I warned you this was coming. All right, a very hypertonic environment. So here's the thing that everyone's gonna hate me for. Maybe not everybody. I like that you guys are a small class. You guys remember how there was a question on the test. Yep, that's where we're going. that had a cell that was 95% water on the inside and was like 50% water on the outside. And a lot of people were very confused and they're like, I don't freaking know. I don't want to worry about it. I don't want to think about it, whatever. And like, would do the best they could, right? So the reason that the outside is hypertonic is because if you have 95% water, how much solute do you have? Salts or something like that, right? 5%. 5% salt. But if you have 50% water on the outside, how much do you have solute there? 50% solute. So that's higher solute on the outside. So that's why it's hypertonic on the outside. Does that make sense? I hope that made sense, but I know everybody wants to be over it. I get it. We got this whole other test to worry about. Why are you going back to the old one? All right. So anyways, so it's important though to understand at least what hypertonic is for this situation, how it creates osmotic pressure, osmosis. And the pressure is just caused by sucking out the water from that cell. If you are packed in salt or sugar, it's going to suck all that water out of you and it will cause plasmolysis. And some bacteria will die from that. But we use high sugar in our jams and jellies, and we also will use salts in order to pack that in there and prevent the growth of bacteria. They can't multiply in that situation if they're in the plasmolysis, but obviously this isn't going to sterilize, right? So here were all of our physical methods, all of them now. I've had them listed out here. I added in the osmotic pressure. So now that means we are ready to go on to the next category. Nope, not going to ask questions, don't worry. But it's probably, I'm thinking of doing it like this way from now on. Like if I call your name, you can pick a question and then whoever's last, unfortunately, gets stuck with whatever the last question is. But it makes it a little bit more interesting, I think. But hey, we'll go through them, okay? So define sterilization, kill everything, right? Can boiling sterilize? You guys remember the answer to that one? Yeah, it cannot. Thermal death time. I'll help you out with this one because this is a term that takes a little bit of time to learn it, but thermal death time. You are putting something at X temperature and seeing how long it takes to kill everything. So you're looking at the time. What's the time that it takes? All right, number 4, which type of radiation penetrates surfaces or whatever? Ionizing, right? That's the dangerous one. And name one method that sterilizes. Well, I kind of just answered it because it was the answer to number 4. You could say ionizing. It could be the autoclave. It could be incineration. There's a bunch of them we could go back to and go visit. But yeah, that chart from way earlier, our second slide or whatever, is really good for that. So now we're going to get into the chemical control. Yeah, okay, we're good. I don't care about you knowing that they could be solid, liquid, or gas because I feel like that's kind of an obvious thing anyways. Cool, it could be all different kinds of chemical states. What you should probably know is that a lot of times if they're in liquid, they're either aqueous or they are tinctures. And an aqueous solution speaks for itself. It's made with water. A tincture is if it has any alcohol in it. Sometimes all alcohol it mixes with, sometimes it's just a little bit. All right, so what we want is the same basic things that we already talked about from our things, but you want it to be rapid in very low concentrations, ideally. You want it soluble in the things that you normally use. You want it to be broad spectrum. Obviously, you want it to be microbicidal, not microbistatic, because you don't want these guys coming back, ideally. You want it to penetrate if you can. Then you want it to be resistant to the presence of organic matter, non-corrosive, non-staining, sanitizing, deodorizing, and affordable. So this is like the ideal chemical, but that obviously doesn't exist. So let's go over our different levels of these chemicals. We have, no, that's my rings. We have the high level germicides. These guys kill endospores, which means sterilizing. And what types of items did we need to sterilize are critical ones. So critical items we treat with high level germicides. If they're semi-critical, go figure, it's going to be intermediate. And obviously, if this is a different category, it's not going to sterilize. And then we have our low levels, things like furniture and the equipment around and stuff like that. But really definitely not going to sterilize. It's going to kill just some of the various groups of the things, but it's enough for those non-critical items. So low level, the non-critical. Okay, so what do we have to think about if we're choosing for our chemicals? Again, this is the same stuff as we already looked at. This is what our microbes are like, the material that we're treating, the degree that it's contaminated, how long we're going to expose it, and then the strength and chemical action of the germicide. We already brought up the IV situation with the gas and like what was ridiculous there, but would you want to prep your patient's arm with a chemical that had to sit for 30 minutes? So that's referring to that time of exposure. It's just nuts. You don't want to do that. And of course, also you want to ask, is it safe to use on the patient's skin? If you have to use it in high concentrations, that could be a concern. All right, so let's talk about how they work. Turns out it's the same as the other one. We're talking about proteins, nucleic acids, cell wall, and cell membranes being, treated or whatever. Whether we're breaking down or preventing the cell wall from being made, sticking in between the cell membrane and preventing it from being a cell membrane working and holding things in, or stopping nucleic acids from being made, or proteins. It can even denature those guys. So it's the same thing here. Here's all the chemical types that we're about to go through. Halogens, phenols and phenolics, alcohols, oxidizing agents, detergents, metals, aldehydes, gases, and acids and alkalis. This is where I would say, and what I did when I was making my notes. As you're going through and writing down what you need to in order to understand this, have your study sheet next to you or near you or something, so you can start, oh yeah, I got that one. I understand that one. I understand that one, at least for now, right? You wrote it down, made notes to make it make sense. This is really a good one for that, because there's a lot of info on these slides that you don't necessarily need to know, okay? Okay, so here's all of those. I'm ready. We're gonna do it. It's gonna get crazy. All right, halogens. Let's start with those. Might sound like a weird thing, but it really isn't. Here we're talking about fluorine, bromine, chlorine, and iodine. All of these commonly used. So we know fluorine is used like to be added to your toothpaste and your drinking to help with keeping your teeth relatively, I want to say keep the stuff from growing on them as best as we can. Bromine, that's often used in hot tubs to keep it sterile. We have chlorine used also like hot tubs and pools and stuff, but in a whole lot of other things. We have a whole lot of cleaners that will have chlorine-based compounds in them, a ton. In fact, like it says down there, chlorine is one of the main chemicals recommended by the WHO for cleaning and disinfecting for COVID. And it's one of the most widely, it says active ingredients in 1/3 of all antimicrobial chemicals. Most of that's chlorine. But we also can use iodine. We know this, you can prep skin with iodine as well. If it's that orange stuff, that's what I'm talking about. So these guys are microbicidal and sporicidal. So if you use them at the right concentration and the way they should be, you could potentially sterilize. Let's start with chlorine. It can slowly kill endospores. So here we're talking about you would have to expose it for a little while to the chemical to achieve, proper sterilization, but it can do it. This will create something called hypochlorous acid. Yes, hypochlorous acid that denatures enzymes permanently, and that's what leads to the killing. This includes hypochlorites like bleach. Bleach is a chlorine compound. So I know people use this, right? This is super, super commonly used everywhere, hospitals and homes, restaurants, everywhere. Hypochlorites like bleach. And chloramines we add to drinking water and to treat wounds. Next is iodine. These can slowly kill endospores, also the same as the other one, so it can sterilize if you use it. Free iodine is one version, and we don't really use that anymore. Then we have iodophores, which are tinctures. Yeah, I don't know what else to say about it. It's not really very exciting. Examples of our iodophores are going to be one thing. Betadine, yes, that's one most of you guys know probably. You work in a hospital anyways. Betadine, povidone iodine, and isodyne. All of these are pretty commonly used as antiseptics on the skin in whatever situations. If you go to donate blood and you are allergic to the compound that they usually use, which we'll get into later, if you're allergic to it, they'll give you iodine. Can you be allergic to iodine? Yeah, you can be allergic to that too. Okay. So it's not a lot of them, like it says, a lot of them are banned now because they are kind of dangerous, cause damage. So here we go. Here's my notes so far. The chlorine with the hypochlorites, which is bleach, and it can sterilize if you use it correctly. I made a little note about fluorine, but it's not on our study sheet. And the iodine. Cool. Next is phenol. I'm not going to go too much more. I think it's only a couple more of those, but anyways, phenol is pretty toxic. It's pretty dangerous and you shouldn't be using it around people. But yes, it says some, so we know it doesn't sterilize. Okay, so phenols do not. And you might be thinking, yeah, cool, phenols, I don't know what that is. You probably are going to recognize one of them at least. Okay, so they're toxic. Even the phenolics are, they're still dangerous and they're derived from phenol. Phenol originally, so it's still the standard that we all compare to. There's a reason for that though. And like it says, we only use it really in cesspools. That's a thing still. Like, I don't know what a cesspool really is anymore other than like in ancient Rome or something. But cesspools, animal quarters, they're just too strong to be used around people. So phenol, it was originally called carbolic acid. Carbofuscin is based off of this and it has that smell that phenol has, that chloroseptic spray / old school Carmex smell. Anyways, phenol is what Joseph Lister used when he was the original person to sanitize his surgical suite or whatever, and started introducing that. So he used phenol for that, but it is toxic and irritating. The phenolics that are based off of that, they will destroy vegetative bacteria, fungi, and most viruses. Again, they don't sterilize. They can be toxic as well. But there's some that are still used that are less toxic than the others. And those two are triclosan and chlorhexidine. Triclosan, which is apparently also dichlorophenoxyphenol. Cool. You guys want to say that word in your presentations? So these are disinfectants that that would be used on, like, I say antiseptics even, really, and it even was used as antibiotic in some circumstances. But when I was growing up as like a kid and a teenager and everything, everybody's hand sanitizer, like these, like just normal hand sanitizer. You go to Bath and Body Works, the hand sanitizers, the soaps that they had, everything had triclosan in it, everything. So, oh, extra killed the bacteria. It's great. And it was until we started finding it in water sources and in animals and all sorts of stuff. So that's why it kind of had, and it also led to microbial resistance as well, which is a huge problem, right? So we only use it in healthcare settings now, and it's rarely used. Then we have chlorhexidine, which if you don't know it up front, you probably know it as Hibiclens, Hibitane, or Paradex. This is like a sudsy detergent type of phenolic that we use this for surgical scrubs, surgical prep on the patient themselves, and other medical uses. But you can get chlorhexidine as a mouthwash. You can get it over the counter. Hibiclens is available over the counter. All of these, I mean, it's pretty tame, but you can become allergic to it, and you don't want to kill off your normal flora. Yeah, I was gonna say it's pretty commonly used on people.
The vet setting too.
Oh yeah, I didn't know about that. Yeah, that's interesting to know. I didn't know that. Oh yeah, the fungi, yes. But yes, so chlorhexidine, commonly used in all sorts of medical or vet or apparently dental for all this stuff, for maintaining, you know, as an antiseptic. I should just say it's an antiseptic. It's very effective. You can use this for treating also, like if you have skin infections or skin sensitivities, even issues with like boils and stuff like that, you can use this to bathe, and is even recommended. You can even bathe in bleach. I know that sounds weird, but it is apparently anti-inflammatory as long as you are only in it for like 10 to 20 minutes, and you have to dilute it a certain amount. So if you guys do it, dilute it, look it up properly. Don't just listen to me and just do it. Don't pour just like a whole bottle of bleach and like sit in it. But yeah. Anyways, I don't know, but whatever. yeah, chlorhexidine is not going to sterilize. We kind of already talked about what it does, and you don't need to know how it kills. That's not in your study sheet. We talked about these. We talked about this. We talked about it has low toxicity. Obviously, it would have to if we keep using it on people. This is what they use if you donate blood. The normal, like clear thing that they sit there and rub your arm with, that clear stuff is chlorhexidine, is what it is. And if you're allergic to that, then they'll use the iodine. And if you're allergic to both, you can't donate. So it's pretty much how it goes. I don't know if you guys know this, but the reason they sterilize like that, or sanitize, I should say, like that, but when you go donate blood, is because the needles, if you've ever watched, the needles are ridiculously big. They're like 14 gauge needles. And that's like bigger. My nose ring is 16. So for the smaller the number, the bigger. So they're bigger than that. And that talks about the bore, not the whole needle, but just the hole inside. That's why they're putting this huge hole in your arm. And then you have to sit there for like 10 minutes with that. And so they want to be sure you're nice and cleaned. And that's why they use stuff like that, and not just alcohol. Like when you go get your blood taken at DLO, they're just going to use alcohol because they have those nice, 20 gauge needles. Alcohol, this is not, again, most bacteria, okay, automatically not going to sterilize. We know it. This is ethyl and isopropyl alcohol. Really, those are the only ones that we are going to use effectively. And 50% alcohol or higher levels will dissolve membrane lipids. And we already mentioned that it affects the cell wall. So now we're affecting cell wall and the cell membrane. So that's pretty good. But of course, these evaporate so quickly, it can not be super effective at doing its job. And then it can affect your nervous system, the vapors can. So again, ethyl and isopropyl alcohol are the ones that you're really going to be using. But when you are treating or trying to get rid of things, you want to get rid of as many as you possibly can. Turns out that 70% alcohol is more microbicidal than 100%. And that is because the water that's present along with that 70% helps coagulate proteins. So now we've got cell wall destruction, cell membrane destruction, and coagulating proteins. It's pretty good. It's kind of surprising that it doesn't, sterilize. But yeah.
But it was called like straight up vodka.
Yes, yeah. And so yeah, there's a reason why they pick ethanol for that too. But yes, so absolute alcohol, it will dehydrate the cells. It has that going for it, but it isn't a protein coagulant. So typically you're going to want to reach for 70. So you guys learned a thing. Ethyl alcohol, we usually use this at 70 to 95%. It is usually going to be used in our hand sanitizers now because it doesn't evaporate quite as quickly as the isopropyl alcohol. And it's usually a little bit more effective on like hand sanitizer application, like the rate of evaporation and how it interacts with the skin. If you're rubbing isopropyl alcohol, which is what most hand sanitizers used to be, it can really dry out your skin pretty quickly, and that just makes you more susceptible to infections. And so, it used to work fine if you were just using occasionally. Now people are using it so often, or at least they definitely were during COVID, that they wanted to switch to ethyl alcohol. Other than that, you might have heard me, one thing you didn't hear me say, I should say. One thing you didn't hear me say is that ethyl alcohol works better, because it doesn't. Isopropyl works better. Isopropyl works better. It's more microbicidal and it's less expensive. So that's the choice if you're going to be cleaning things. But if you're exposing your hands to it pretty regularly, you might want to go with ethanol for your own integrity of your skin. Cool. So here's where we are. Look how much more we have to go. I know. Oxidizing agents. So this isn't so bad. This is actually, we're going to be more familiar than you think it's going to be. So remember, I have Leo says GER, right? So lose electrons, you are oxidized. But if you are oxidizing something, you are making it become oxidized, right? So you're making that thing lose its electron. That's what it means, okay? So oxidizing agents. These can kill endospores and other microbes. Interesting. So these can sterilize. Now, how about this next part? Hydrogen peroxide. It's pretty interesting, yeah? It's going to be in this category as far as that. Now, of course, it's going to fall into oxidizing agent when I say hydrogen peroxide. Okay. That makes sense. But for it to be falling in this possible sterilization category, pretty interesting when most people reach for alcohol to treat things. It does, all of these cause free radicals to form, and they are only sporicidal, so only going to sterilize in very high concentrations. Okay, so if you're using it at the lower concentrations, they're not going to sterilize. So with hydrogen peroxide, we use it at 3%, which is not high. So the brown bottles that we're so used to seeing, they have to be in the brown bottles to protect from the light. That'll cause the H2O2 to break down into its constituents. But anyways, you can disinfect with it. And it's pretty effective at that. And we'll treat these mostly with anaerobic bacteria, because if you are something that lives in the air, oxygen, we need oxygen, right, for our aerobic processes. So we have to deal with potential toxic byproducts associated with oxygen. So we have to have some system in place to get rid of them or protect our cells or whatever it is. But the anaerobes, they don't ever see oxygen. So the products related to oxygen that can be damaging, they don't have any defense against it. So this is mostly for treating anaerobic. And so what I mean by that is deep tissue stuff. You do like a wash with hydrogen peroxide. Is it going to help treat things that are at the surface? It can. There's no harm in using it, and it's pretty safe. You can use it as a mouthwash too. Anyways, 35% is what we would use in a sterilizing cabinet, which is effectively almost like an autoclave, but with vapors of hydrogen peroxide. All right, so with hydrogen peroxide, the way that we manage to deal with it is we have an enzyme called catalase. And I know someone brought up in my last class, I can't believe they're paying attention that much, that the equation isn't balanced. Cool. Okay. I know it's not. I don't care, and I couldn't possibly be made to care. But this is essentially what it does. It's going to have H2O2, which is your hydrogen peroxide. So you have a hydrogen per oxygen. That's why it's called that. So hydrogen peroxide, when we have catalase, our enzyme there present, it will convert it into H2O and oxygen. Easy enough to understand it, gets rid of it. Now we're safe, we don't have to deal with it. But when you guys use hydrogen peroxide, do you ever notice those little bubbles, the foaming or whatever that you can get? Not all the time, but sometimes we see them. That's the oxygen that's being made by the catalase. Usually it could be in the bacteria or it could be in your own cells because you have it too. So it's pretty interesting to think about that. And we'll test our own bacteria that we work with for that later on. So yes, it can be sterilizing at high temperatures. Obviously, we can use it as an antiseptic at lower and a disinfectant. We can use that for items. Remember, that's going to be for non-living surfaces. And we can sterilize at high concentrations if we vaporize it. We can even do whole rooms like that. Next, we have the detergents. These are surfactants, okay? And that's on there too. Again, some doesn't sterilize, okay? We want cationic molecules for this one. Now, we talked about how you have your phosphate heads. This is your P, your phosphate head, and this is your tail down there. And that's a negative charge, and it's hydrophobic at the bottom. Cool. These guys have a positive charge and a similar makeup because the negative likes the positive a little bit more. It will associate with it a little bit more. So it makes it more effective. So quaternary ammonia compounds, which are based on ammonia, NH4, which will have a basic aspect, so positive charge to it. Anyways, it'll get into that membrane. I don't know if I put the picture back up here. I don't remember if I did or not. Oh, this one doesn't want to work. Cool. Okay, whatever. Anyways, but yeah, breaks into that membrane and loosens it up just like what we saw before. So here's a picture of some surfactants that'll help disrupt that membrane permeability. Again, these are amphipathic because surfactants are amphipathic. And they're all like these ones, again, nitrogen. And this one down here underneath it, nitrogen. So the one at the bottom is benzalkonium chloride. It's pretty commonly used antiseptic as well. It's like what they gave me when I got my ears pierced at Claire's or whatever when I was a kid. But yeah, so it'll have that positive charge and then some rando tail on the end of it to help it get into the membranes. We call these, again, these quaternary ammonia compounds, we call them quats, and you probably use them a lot more than you think that you do. A lot of soaps are based on this concept, but they're not very effective killing. We have Pseudomonas bacteria that'll grow even in soap dishes while your soap is sitting there. So they don't care. It's not super effective. But if we combine this that helps weaken the membranes and mechanically removes with something like a phenol or even iodine, that's usually what you're going to see when you have a chlorhexidine prep. It's going to have something else behind it that's helping, or like Hibiclens. We have that detergent in there to help it. Next, we have heavy metals. These guys don't sterilize either. And you can develop a resistance to them. They can be toxic and you can be allergic to them. The two that we use the most are going to be, come on thing, catch up. Mercury. Yep. And then silver. And then silver. Both of these are used as antiseptics. Yep. Y'all didn't know that, did you? So we can use certain organic mercury tinctures on your skin as antiseptics on living, parts of your body tissues, or we can use them as preservatives in cosmetics, ophthalmic solutions, and other things as well. Probably didn't even know that. Putting contacts in your eyes with mercury on them. Not all of it has to have that, but some of them do. All right, so we already said about the mercury. Cool, it's a preservative and an antiseptic. Silver, though. We have silver nitrate we use topically. We have silver sulfadiazine. We use this for burns. If you ever had a burn, usually 2nd degree or more, you expose like the layers underneath and there's concern of those becoming infected, they'll give you that to treat it. I only know because I had to have it. Pure silver, they incorporate it into catheters. Colloidal silver, you can drink it and stuff. And I don't know, people think that that's so good for them and then they turn blue because it turns you blue. Anyway, silver ions that can be incorporated into hard surfaces to make them antimicrobial, or like scrubs that you can buy that are antimicrobial often have silver in them. So here we are, we're caught up with our metals, and I made some comments about how they work. Next are the aldehydes. They can kill endospores, finally, and other microbes. Yes, sterilize, potentially. All right. CHO group is what dictates what is an glutaraldehyde is one of these. So it's an aldehyde that can be sterilizing. We also have orthophthaldehyde. It's just like there at the end of the sentence. I don't know why, but it is. But it's very expensive. So let's go through them. Glutaraldehyde, it can sterilize. It's kind of toxic and can be damaging to tissues. It sterilizes materials that would otherwise be damaged by heat. Like instruments or whatever. Then we have formaldehyde. Usually we might see this in the aqueous solution form. This will be formalin. It'll disinfect. That means no to sterilizing. So formaldehyde doesn't sterilize, but we use this as an embalming fluid. It's pretty effective for that. Last, we have that orthophthaldehyde. Yep. I want you guys to say that. Oh man. All right. OPA. It works like glutaraldehyde. It's faster, but it's not as effective and it's more expensive. And it's not as irritating though. Hey, so it just depends on what you're using for what there. All right. The gases, we have ethylene oxide. This will be a gas that kills endospores. So this is really interesting, right? It's just a gas that can be completely sterilizing. It's very effective. And it's vigorously functional to destroy, DNA and proteins. Great. It's a sterilizing gas. Wonderful. So why haven't we heard about it? Why don't we know about it too much? Well, it's explosive and it's a carcinogen and it causes damage to tissues. So that's a problem. And I mean, it is explosive. So they have to have some sort of situation involved in order to deal with it. So why would you ever use this though? If it was explosive, you can treat entire rooms with this and it's super effective. And then if you do that, everything in there is going to be sterilized. So we call that a chemiclave. It's like an autoclave, but with chemicals. So you have some way to deal with it afterwards. We have chlorine dioxide. This is what we would find in drinking water, wastewater, when we treat it, food processing, medical waste, and things like that. We used chlorine dioxide to get rid of anthrax in 2001 when the Senate was attacked with that bioterrorism incident. And so Bacillus anthracis, and if it's Bacillus, that means it has endospores. So that means that it also sterilizes. And then we have the acids and the alkalis. Again, some bacteria, so we don't sterilize. And then we have the way that it works is by a pH alteration. Our proteins rely very heavily on their hydrogen bonds for their structures. You interfere with that with more or less H pluses, protons, then it can cause some serious issues with the proteins and how that they are going to fold and therefore how they're going to function. Ammonium oxide and ammoniums in general, all these ammoniums, they are basic. They are alkali. Alkali is basic. And then we have the organic acids. These include acetic acid and lactic acid. Lactic acid is, what is happening? Oh, thanks a lot. Tell me I have lab in 15 minutes, thanks. Acetic acid is vinegar. So that's just another name for that and why it's effective. So it's a nice, tame, non-damaging, easy to use acid, very kid-friendly, animal-friendly around the house for cleaning things up. Doesn't sterilize, but do you really need things sterile? But yeah. So here is my final, come on thing, sheet for the chemicals. This is how I would like maybe organize my notes as I was going along with the lecture or something. But this is my ultimate notes, okay? Hey, there's some chemicals on here you probably recognize. I'm not going to go over them right now. Oh, and I didn't open up the other thing. Great. So that's it for that. And then I summarized all my notes in this. And these are posted online, by the way, if you guys wanted to look at my notes afterwards and see what I wrote and how it works. Because yes, these notes just won't keep up with what I'm doing. These notes literally cover every single thing on that study sheet for chapter 11 anyways. They do. So there's that. Plus, what I recommend is after you make your notes that are just what you need to check off those boxes on that study sheet, then afterwards go back over your notes and then reorganize it into something else. This was supposed to be a lot cleaner and it didn't end up being. You could do it again if you wanted to, but reorganize it will also have you like rethinking about how they relate to one another. Get you thinking about it. My recommendation, take the notes that are just what you need, then right after class, reorganize it. If you need to look up something that you didn't understand, look it up and incorporate it in there. That's all you should need to do for chapter 11. You put that down until the exam. When it gets to the exam, you look at that sheet. That's it. That's how you study. Y'all watching videos, y'all reading textbooks, you rewriting your notes. Stop it. You're just confusing yourselves, especially whenever you're tired and you're stressed and you're trying to get all this information, cram it in all at once. Tell me that's not the reason why you guys don't do well on the exams. Tell me it's not. So if you are doing, poorly on exams, I don't know who is. I don't remember. If that makes you guys feel any better, I don't think about it that way. But yes. So anyways, this is my recommendation on how to do that. These are on Canvas under chapter 11, and it literally says like professor's notes or whatever. I'm not going to have these for every chapter. I just wanted to put it together so you guys could get an idea of what I'm talking about with that. If you ever want any help with making notes or whatever, then let me know. But yeah, if you guys don't think that this would work, take this one sheet. This is my last one, my reorganized one, and look it over, see what's on there, familiarize yourself with it, and then use that to take your quiz or a Kahoot or something and tell me it doesn't work. Because this doesn't need to be all the BS that people are doing. It's too much, okay? And if you do this up front, take good notes. I don't need this. Take good notes and then reorganize it some way that makes sense to you, right? Then when it comes time to the exam, and all you have to do is review this and maybe make some flashcards for some of the terms that didn't stick, right? And that's it. You don't need to be sitting there for 6 hours So many students come and be like, I spent 5 hours writing these notes from rewatching the lectures 3 times and I didn't pass. Yeah, because you're just listening and writing down words and you're not actually understanding it. If you go through to take the time to make sure that you're understanding it and how it relates, that's going to save you so much more time. And if you do it with just this one chapter at a time, you're way more likely to succeed with one chapter at a time than trying to cram all 6 of them in, or God forbid the final when you have to do all like 25 chapters. Makes sense? I'm just trying to help you guys. If you don't want to use it, that's fine. It's absolutely fine, but it is there for you guys to use if you want to. Now, let me see. Let me put this back on. Okay, well, why can't I stop with this? Zoom, where are you? Stop sharing. All right, we're going to put on our little Kahoot for our lab stuff. That's what I'm going to do. We're going to do it like a normal person on this like computer. Okay, cool. Yep, you asked me about cookies on every single thing I click on. You guys have any questions about anything while I'm doing this? So I can only talk a lot, and I know that chapter 11 is just a lot of slides, but it's really mostly terms, luckily. Memorization stuff, that's not too bad. When we get to chapter 15 and 16, because it is immunology and they like play in with one another, I might do another like study. I don't know about study session. We might, if there's enough interest, but I mean like walking through how I would create, or at least like showing you what I would do for studying for that, because they both relate to each other. You can't just have one of them. My password. Oh, crap. Did I type it correctly? And this won't take long. We'll be done early for sure. I know I say that sometimes and it doesn't happen, but that absolutely will happen with this. Lab. So midterm. We're gonna do it the normal way or like the boring way, but that way I can at least talk you through some of these. Yeah, we'll talk you through some of these questions. And I think I have this posted